if you say the globally, that where it's very difficult to degrade the plastic. So he will be highlighting some of his research points where his lab is working on the role of biofilms in the plastic degradation. So with these words, I invite Professor Antaresh Kumar to deliver his talk.
So, good morning. Today we have selected a new topic, that is the role of biofilm in the plastic degradation. So far you have discussed different aspects of biofilm in pathogenesis, in other aspects like food processing. So today's topic is related to that role of the microbial biofilm to degrade the plastic. And all know that plastic is the major problem worldwide, which causing a different kind of pollutions in our ecosystems. So key questions came into the mind that when we deals about the plastic, how big the plastic for problem is, how much plastic is we generally generate, and how much plastic we consume, and what are the management of the plastic, whether the plastic which we produce is actually recycled, or how do we manage the plastic after this. Second question is how much plastic we recycled, the plastic which we generate, that what amount of plastic we recycled and reuse it again. And the third and most important point is how do we manage the plastic pollutions or plastic waste which we generate. So the today's topic is related to the third questions which we have in the mind, then how do we manage the plastic waste. So if you talk about the plastics, overall the worldwide plastic we produce around 415 million metric tons generally plastic generates all over the world. And out of this 415 plastic, 360 million metric ton plastic we generally trash into this, generally trash it, which is approximate 87% of the total amount we generate. So this is a major problem in the plastic, we use it and throw it into the trash. And the third one is how much amount of plastic we recycled worldwide. So approximate 17% of the total amount, which is equivalent to around 62 million metric ton we recycled, which is 17% is not that significant number which we recycled. Thereby the most of the amount of the plastic undergoes to the waste that causes the problem into our ecosystem. And the most important is this, how do we manage the plastic after use it. So plastic management, plastic waste mismanaged around 82 million metric ton, which is 23% of the plastic we doesn't care about it after use it. So that causes the problem in our ecosystem. Problem major in the mismanagement of the plastic, we use it and throw it here and there. That causes the problem and causes the pollution in the environment. So these are the different countries worldwide produce or consume the plastic, and we stands in the third position after China and US. And the contribution in our terms of the global, that is 6% we generally produce the plastic all over the plastic generates around the globe. Plastic after the use we generally throw in the different areas, particularly the primary site where we can throw the plastic is lands. And if you look at the data, approximate 50% of the plastic which we generated generally fell into the lands, that is approximate 178 million metric ton plastic, generally landfill plastic, which creates a waste or pollution or generates the gases after degradation and disturb our ecosystem. Along with this landfill or soil, We also throw the plastic into the aquatic systems, and if it is look at this UNESCO data, approximate 100 million metric ton plastic is already present in the ocean, and per year we generally throw this 17 million metric ton plastic into the oceans, which disturb flora or fauna of the aquatic systems. How this aquatic system get disturbed by the plastic waste? So the plastic which we throw into the aquatic system gets converted into the microplastic by different means like biofilm, microbial biofilm, some sewage treatments and some UV degradations. And ultimately this plastic converts into the microplastic, will be ingested by either the some of the fishes or some of the aquatic organism, and also adhere to the algae or some plants present in the area in the aquatic system, and this microplastic ultimately goes into the food chain, in the human food chain by different ways. Not only in our case, it also goes to the some of the the birds which we can generally attack or eat or take this fish as a food. So in that way the plastic which we throw into the aquatic system ultimately goes to the human as well as also goes to the different species and cause the pollution, and it shows the detrimental effect onto the human metabolic system as well as their bird metabolic systems. So these are the different worldwide studies. There is a study that they have tested the different freshwater rivers, the plastic content present in that rivers. So they have checked in the China has this, they have taken the sample of this particular reservoir, and they have found there different type of plastic like polystyrene, polypropylene, polyethylene in different amounts. Even though in the Vietnam, Portuguese, Brazil, and look at this is the India, northwest India, particularly the Ganga or Yamuna is also get polluted with the plastic, having a different type of plastic presents that affect the overall flora and fauna of the water system in India as well. What are the impact of the plastic if it contaminated our land, aquatic system or air? The plastic, if it is present in the soil, can affect the soil fertility, can deteriorate the soil quality, and as well as if it is the plastic is present in the soil, after this the degradation it produces some kind of gases which is toxic to the environment and participate in the global warming. In addition to that, the plastic underleach into the aquatic system, that may also affect both the productivity and the growth of the aquatic organism as well as the flora and fauna of the aquatic system. So overall the plastic, if it is present in the soil system and the plastic waste present in the air as well as in the as well in the aquatic system, will produce a detrimental effect all over the ecosystem. So if we talk about the plastic, majorly are two type of plastic, as you know. One is the thermosetting plastic, another one is the thermoplastic. Thermosetting plastics are those which can heat it once and then it converted into the hard structure, hard substances which cannot be remolded to if we reheat it again. So such kind of plastic is called as thermosetting plastic, and these are the different example of thermosetting plastics. That urea formaldehyde is used as a thermosetting plastic, which are used in the electrical fittings, makes and knobs and some additives. Polyurethane, examples of automobile interior, make a foam insulator layers, is a polyurethane. Phenol formaldehyde, bakelite, that makes a electrical switching, machine parts of the electrical systems, and also epoxy resins which also used in the casting and encapsulation adhesive and also other materials. Bakelite is also used for the electric switches and other electrical versus cookwares. So these are the different examples of the thermosetting plastics. And next category of the plastic we can is that it's thermoplastic. Thermoplastic is the main one which we generally use in our daily life, like polyethylene, polystyrene, or some other type of plastic we generally use in the daily life. So these are the example of the thermoplastic, like polyvinyl chloride. We generally use the pipes we made up of this PVC, credit cards, tiles, and also pipes. Low density polyethylene, that is LDP, that also used in the garbage cans, frozen foods, and plastic wraps is also made up of this low density polyethylene. High density polyethylene is basically the milk bottles, bleach type bottles, sparks, and other articles are made of this high density plastics. Polypropylene, the Tupperware, car parts, yogurts containers are made up of the polypropylenes. Polystyrene, the egg cartoons, disposable dinnerwares. Nowadays we generally use in the marriages the plates which are basically made up of the polystyrene will also produce the pollution after use it and throw it into the soil. Polyethylene terephthalate, which forms the food tray and beverage bottles, these all are the example of the thermoplastic which contributes majorly into the plastic waste in our daily lives. So if we look at the Indian scenario of the plastic we generally use, so overall the India produces the total 25 million metric ton plastic per year, and the total waste we generate around 6.6 million metric ton, which is almost the 26% of the total amount we generally produce of the different type of plastic. And the mismanagement of the plastic in India is the very worst condition. The plastic which we use it generally throw it, and in that case India is the top position to mismanage the plastic. After use it can throw here and there, and very less amount of plastic gets recycled. Around 3 to 5% plastic we recycled and reuse it again after the once time. And the most striking data is Delhi is the highest generator of the plastic waste. It is the only city all around the India which produce the highest number of the plastic waste, that is around 1100 metric ton per day. That much plastic is produced by Delhi itself. So to think about how to manage this plastic, so we generally consume India level, there are different type of plastic we consume in the different ratio. For example, we use in India polypropylene we use approximate 33%, polyethylene we use approximate 30%, and 70% we use the PVC for the pipe and other purposes in India. And rest that contributes around 18 to 20% in the total of the plastic we use. The major problem in the plastic is the conversation, because the conversation or the decomposition of plastic is very hard. It takes time, and in the composition that the plastic which is the convert first into the microplastic or nanoplastic produce the major pollution that ingest by the organism like plant or aquatic system. So the recycling and conversion of the plastic can be done will be three different means. It can be done either by the chemical method can be used to convert the plastic into their last product like monomers, or can be used that monomer to recycling purposes. Another one, we can use the biological approach. The biological approach can be used, some plants can use a microorganism, fungi that can convert this macromolecule or polymolecules into the simpler form. And third one is the hybrid mode. We can use some chemical or environmental or thermal condition or physical condition along with the biological condition can upscale the plastic recycling. The upscaling of plastic recycling is related to we can reuse this plastic some other purposes, and I mean the plastic can be used for the second time or multiple time by upcycling. So as compared to the chemical method, I am not going to discuss in detail. The pyrolysis is the one way we can convert the plastic into the gaseous form. Depolymerization and gasification is another method of the chemical methods. We are more focusing in this biological method of the plastic utilization or recycling or decomposition or conversion of plastic into the simpler molecules. So if you talk about the biological method, first microorganism came into the mind because that is the natural process that microorganism tends to adhere onto the surface of the plastic and they change the plastic is basically a hydrophobic in nature. So for microorganism, either bacteria or fungi or some other organism, they tend to adhere or interact with the plastic, will first try to convert the hydrophobicity, reduce the hydrophobicity by changing some functional group present in the plastic. So microplastic, basically a microorganism biofilm first adhere to the plastic and convert the plastic into their less hydrophobic structure. So once the microorganism adhere to the plastic surface, it tends to release some enzymes, it tends to make some extra polysaccharide, polymeric substances which can change the functional group present in the plastic, and in that way it can change the reduce the hydrophobicity of the plastic. So microorganism biofilm play a critical role for the adherence. Not only adherence, but it also has a role into its degradation by changing the hydrophobicity of the plastic into the hydrophilic nature. Once the hydrophilic is nature, then it gets digested or decomposed by the microbial enzyme. So this is the overall mechanism of the plastic, that microorganism biofilm tends to help in the adherence to the plastics. Once the microorganism biofilm adhere to the plastic surface, it tends to change its hydrophobicity, reduce the hydrophobicity by converting the polymorphism functional group into the hydrophilic groups, and these hydrophilicity increases that helps to facilitate the acting the enzymes present in the biofilm region to degrade the plastic into the simpler molecules. So microorganism quickly form the plastic, and the microorganism grown in the plastic term is called as the plastospheres. So the plastosphere microorganism forms the plastospheres that acts as a miniature ecosystem, that acts as the small ecosystem that helps to degrade the plastic by releasing a different type of enzymes. So how this biofilm formation occurs onto the plastic by different microorganisms. So there are different steps are involved in the biofilm formation. First step is the adherence or attachment of the microorganism to the plastic. So this is a microplastics, and these are microorganisms that they adhere, and initially there is a certain molecules expressed by the microorganism, like biofilm is active, certain EPS molecules is also synthesized that helps that facilitate the microorganism to the adhere onto the hydrophobic plastic surfaces. So first step is to the attachment that provide the hydrophobic nature of the biofilm of the microorganism, facilitate to adhere or bind or attach to the plastic surfaces. The binding of plastic surfaces depending upon the nature of plastic, whether it's the polyethylene, polystyrene or some others. Another nature is how this EPS is releases, amount of EPS, amount of enzyme is releases that provide the interaction between the plastic to the microorganism. So after the microorganism gets attached to the surface, the next step of the biofilm formation is biodeteriorations. As the name suggests, there are certain number of enzymes is releases after the attachment of the micro to the plastic surface. These enzymes tends to secreted extracellularly that can adhere, that can converts the plastic into the different monomers or different forms. Particularly in that particular stage tends to start the hydrophobicity alteration. I mean the hydrophobicity tends to reduce by attaching, by releasing some enzymes at this particular stage at the biodeterioration step. After the second step, the third step is this biofragmentation. As name suggests, the polymers of the bioplastic gets biofragmented with the help of the different type of enzymes releases and the long polymer chain of the plastic can be converted into the simpler form by this particular steps, biofragmentation, and these converts into the simpler molecules of the polymers like polyethylene converts to the ethylene or some other related compounds. These similar monomers of the plastic can be utilized by the microorganism for their growth and act used as a carbon source for their growth. So the next step, fourth step of the biofilm formation is assimilation or mineralization. This simpler molecule synthesized by the degradation or biodeterioration of the plastic. This simple molecule can be assimilated by the microorganism that can be used for their growth by different like abiotic or anaerobic or fermentation process of the microorganism required for the growth for the microorganism. Some of the molecules which are not utilized by the microorganism that also be non-assimilated foods we also synthesize inside in the culture. So both monomers which is assimilated by the microorganism or utilized by the microorganism or not utilized by the microorganism will also synthesize during this step. And ultimately this monomer convert to the carbon dioxide or water molecules as a mineralization form. So ultimately the plastic microorganism adhere to the plastic ultimately convert the plastic into the mineralization form like carbon dioxide in the water by getting metabolized by the microorganism cellular mechanism or may also convert some other molecules depending upon the type of plastic. There are different variety of the microorganisms have been reported that can be used for degrade the degrade the plastics. These are the different genus. I am not going to discuss one by one. So these are the list of the genus or these are the different families of the bacteria will tends to degrade different kind of plastic like LDP, polyethylene, polypropylene, polyvinyl alcohol, polyethylene and polypropylene, polystyrene, etc. So there are a number of reports have been published related to identify the microorganism which can adhere to the plastic and degrade it into the simpler form. What are the factors responsible for the plastic degradation? So depending upon the biofilm, there are two types of major factors involved in the degradation of bioplastic by the microorganism. One factor is rate and quality of the biofilm formation, how much amount of EPS is formed, how efficiently cells grow onto the plastics. So all these factors affect the plastic degradation. And second one is the type or property of the microplastic, whether it is the polyurethane, polyethylene or PVC. If it is PVC, it is very hard to degrade as compared to P or polypropylene. So these are the two major factors. One is the nature of the plastic, what is the plastic, what kind of plastic it is, and second one is how what amount of the or EPS or how fast the bacteria or the microorganism grow into the plastic will affect the plastic degradation. In addition to that, certain physiological factors certain other factor will also affect the plastic degradation, such as the temperature. If it is little bit increased, will increase the metabolic activity of the cell, also helps to faster degradation of the plastic. pH is also one factor can affect the plastic degradation, microbial plastic degradation. UV is the another one, and the nutrient condition, limiting nutrient condition may also augment to plastic degradation by the microbe. So in addition to those two factors, some other environmental factors will also help to degrade the plastic by different type of microbes. Let's take the one example of this degradation of polypropylene or polyethylene plastic. So before discuss into the detail, first we'll go through the outline and then go to the mechanism how it works. So this is, let's say, the polyethylene or polypropylene particles where the microorganism is grown attached there, and this is the chain of the polypropylene or polyethylene. So this chain can be altered with this hydrophobic chain converted little bit hydrophilic form. Hydroxyl group is hydroxyl functional group is attached to this. In that way, the hydrophobicity of the plastic gets altered by attaching the microorganism using certain kind of enzymes. So before discussing how this plastic, basically polyethylene or polypropylene, gets degraded, first we need to understand this polyethylene, is the polymer of ethylene, and there are different type of polyethylenes are present depending upon the branching chain. So the least branching chain is the high density polyethylene, which has the very short side chain, few or no branching. Another one is low density polyethylene, which has the significant number of the branching as compared to the carbon-carbon chain. So in addition to the carbon-carbon chains, the low density polyethylene has the higher number of branching carbon chain. So this higher the number of branching, higher the flexibility and the lower the density. This is the relation between the flexibility. So these are the this less flexible, and the low density polyethylene is the more flexible. So another one is the propylene, is the polymer of propylene, this monomer of propane. So there is a polymerization of this particular catalyst can convert into 23 to 35 carbon chain longs. It may be the arrangement of this propene monomer into this range, I mean has the regular methylene group attached. That kind of polypropylene is called as entactic propylene. Maybe the one sided of the methyl group attached there in the polypropylene. Such kind of propylene is called as isotactic polypropylene, which has the one sided methyl group, and some may have the propylene, the arrangement of methyl group into the alternative form. That kind of propylene is called as pseudotactic polypropylene. So if we know, once we know the structure of the polyethylene and polypropylene, so how does the polypropylene and polystyrene gets degraded? So I told then once the microorganism adhere to the polypropylene or polyethylene surfaces, it first tends to increase the hydrophilicity. So this is the polyethylene, and the microorganism adhere to this, then it's form the formation of hydrolyzable groups. So it converts the hydrophobic carbon-carbon in attached to the carbon-carbon backbone of the polypropylene or polyethylene to convert into the hydroxyl group attached by the action of certain kind of enzymes present in the biofilm area of the microorganism, and some can be extracellularly released from the microorganism. Enzymes can be alkane dehydrogenase, cytochrome P450, Weir Wieseler monooxygenases, alcohol dehydrogenases, and aldehyde dehydrogenase. These are the enzymes can first add the functional group to the polypropylene or polyethylene surfaces. As a result, their hydrophilicity increases by the microorganism. And once this is hydrophilicity increases or functional group attaches to the carbon-carbon chain, then under second step is the hydrolysis of this hydroxylated polyethylene or polypropylene. So first step is to hydroxylate the polypropylene or polyethylene surface by the microorganism by acting the different enzymes, the list of enzymes, and after then it gets converted into the simpler or monoform, which can be utilized by the cells for their growth and metabolism. So hydroxylases, which are different kind of hydroxylases present that can facilitate, particularly for conversion of carbon-carbon chain, converts the functional group, add the hydroxyl group there to reduce the hydrophobicity, is the first step for the degradation process, and followed by then the another hydrolytic enzyme acts on the modified chain. So these are the list of enzymes have been reported that used to the polyethylene degradation or polypropylene degradation. I told these are hydrolytic enzymes such as cutinase, lipase, esterase, which has been reported in a different type of microorganism that extracellular releases can form the carbon ketone hydroxyl bond and some other bonds for the helping in the degradation of polyethylene or polypropylene. So I have taken the one example of this particular strain, which synthesize the P450 enzyme, and P450 is the oxygenases that can form the ketone in the hydroxyl group and also involved in the amide bond formation. So this particular bacteria, so there is a report they have grown this bacteria in the different concentration of polyethylene. So this is they have measured the growth of this particular bacteria Bacillus thuringiensis JNU1 that degrade the polyethylene. So what they did, they grown this bacteria in the different concentration of polyethylene, and if you look at the growth, this as the concentration is increases of the substrate, the more amount of growth is observed. So that means this Bacillus thuringiensis JNU1 strain tends to degrade the polyethylene, utilize the polyethylene for the growth. As a result, the growth is increasing. You can easily visualize the turbidity of the bacteria grown into the agar medium. In that media has the polyethylene present in this agar. So this particular strain of the bacteria, which is JNU1, can also decompose the polyethylene into the low carbon monomers. So this is the GC mass spectrum diagram, which you can see the different small molecules. This is decomposed the small molecules with the treatment of Bacillus thuringiensis JNU1 that can show that the polyethylene can degrade it into the different type of small carbon molecules. They have also measured the, so once the plastic is adhered, once the microorganism adhered to the plastic, it increase the hydrophilicity. If it is the hydrophilicity is increases, the water contact angle gets water contact angle reduces. So they have what they have measured in this particular area. This is the non-treated polyethylene film, and they measure the water contact angle, which is around 97 degrees. And once they have treated the same material with the Bacillus thuringiensis JNU1, then the water contact angle gets reduced by from 97 to the 77. It means there is the hydrophobicity of the polyethylene gets decreases. As a result, the water contact angle is decreases with the action of this Bacillus thuringiensis JNU1. They also measure this the nature of action of this Bacillus thuringiensis JNU1 strain onto the. So this is the polyethylene sheets, and this is the treated sheets. They have observed the different magnifications. So when they have treated this polyethylene sheet with the Bacillus thuringiensis, the morphology or the structure of this polyethylene film gets disturbed. It means the polyethylene sheets gets be degraded or utilized by this Bacillus thuringiensis JNU1 strain. Another, they have find out the role, what are the properties of this particular bacteria play to degrade this polyethylene. So they identify there is a cytochrome P450, which is the hydroxylate the polyethylene. I told when in case of the polyethylene degradation process, first microbacterium binds attached to the plastic surface, and it releases some enzymes such as this cytochrome P450, which hydroxylate the polyethylene, and in hydroxylate means OH group is attached to the carbon-carbon backbone. So what this particular cytochrome P450 is involved to the degradation of polyethylene. So they have measured the different type. These all three are the different type of cytochrome P450, but as compared to this and this, the activity of this cytochrome P450 102A5 is the highest activity in presence of this. It means this particular cytochrome P450 hydroxylate the polyethylene by this Bacillus thuringiensis JNU1. And the same thing they have also measured by the GCMS. So they did the GCMS in absence or presence of this enzymes treated with the polyethylene. So this is the treated polyethylene with this cytochrome P450 gets some alteration in this, which is the sign of the hydroxyl group attached, or hydroxyl group emerge into the polyethylene. Next research is related to that can degrade the polypropylene, not polyethylene. So this is the bacteria Pseudomonas aeruginosa WGF6 efficiently degrade the polypropylene. So they have checked another research, they have checked the growth curve of this Pseudomonas aeruginosa WGH6 in presence or absence of the polyethylene. So this is the minimum media and the polypropylene with this particular bacteria, and this is the minimal media with polypropylene with this bacteria WCS6. So as compared to this, WCS6 has the higher growth, although the WZH4 is also grow in the presence of the polypropylene. And next one, this is the only the MS medium with polypropylene, and this is the MS medium without substrate polypropylene. So this particular graph shows that this WGH6 and WZH4 grew in presence of the polypropylene, suggesting that polypropylene getting utilized by the Pseudomonas aeruginosa cells, but as compared to 461 is more efficient, degrade or utilize the polypropylene during this growth. The same they have also measured with the weight loss of the polypropylene. So this is the WZH4 and this is WGH6 of the aeruginosa, Pseudomonas aeruginosa. So as compared to WZH4, WGH6 having approximately 70 to 18% polypropylene weight loss observed in this case, and also the water contact angle is also reduced in case of the WGH6, which is 56, in that case is 76 degrees. So it means this W6GH6 of the Pseudomonas aeruginosa efficiently utilize or degrade the polypropylene for their growth, bind it and degrade it into the simpler molecules which can be metabolized by this particular bacterial strain. Next one is PVC, which is very recalcitrant to degrade by the microbes. Very few reports are available, only 1 or 2 reports where the microorganism can degrade the PVC. So this is the polyvinyl chloride, and there are different kind of enzymes are involved, and this PVC can convert it into their different monomers like alkanols or alkanoic acid. So what are the mechanisms? So degradation of the polyvinyl chloride is started into 4 different steps. First step to removal of chlorine, that is oxidative dechlorination is the first step. So the bacteria adhere to the PVC surfaces and tends to release some enzyme that can dechlorinate the PVC, and that forms the dechlorinated PVC. There are a number of enzymes such as non-heme chloroperoxidase is released by the bacteria, and this particular enzyme catalyzes halogenation using hydrogen peroxide. Also the perhydrolase is also enzyme releases, which per acid forms from acid or H2O to hydrogen peroxide, and acid can form the peroxide. That is the function of the peroxide perhydrolase. Halo acid dehydrogenase is also used, that cleaves this particular bond, CX bond, CCl bond, and releases the halogens and form the hydroxyl group, OH group it achieves. So first step for the degradation of PVC is to oxidative dechlorination, in the removal of chlorine first, so that the chlorine can be present and the position chlorine can be hydroxyl group, ultimately tends to reduce the hydrophobicity of the PVC. Second step of this is oxygenated PVC, rather than that nature of oxygenated oxide group is increases, that increases the hydrophilicity of the PVC, can make a vulnerable for the degradation. So there are different type of enzymes can be induces, like induces hydrolyzable functional group formation, and these are the list of enzyme can be used that increases the hydrophilicity by adding a more oxygenated group, more oxide group to the PVC chains. Third step of this is hydrolysis or fragmentation. After the PVC can be oxygenated, having a more oxygen group, then enzyme can easily attach, because the nature of PVC gets relatively more hydrophilic. And the next step is the hydrolysis of the PVC material. So the function of this, the depolymerize oxygenated deep PVC, and in that case different type of hydrolases can act onto the oxygenated PVC, like esterase, dehydroxy acid, dehydrogenases and lipases. These are the list of enzymes can be used to hydrolyze, convert the PVC into the simpler molecule. And the next one, once the simple molecule gets synthesized, can be used, can be metabolized by the bacteria. So these PVC degrades in the 4 different steps. Different type of enzymes are involved that can first convert the most hydrophobic or recalcitrant this PVC into the hydrophilic nature, and then increase the hydrophilicity. Once the hydrophilicity is increased, then easily hydrolyzable enzyme can act on that to degrade the PVC into the simpler molecules. So this is the one report, one bacteria. This is the only report I got for PVC degradation, that is Klebsiella EMBL1, that can efficiently degrade the polyvinyl chloride. So this is the control cells, which is the polyvinyl that in the medium, and this is the bacteria grown, the Klebsiella, that can disturb the structure of PVC when it is grown onto this surface. So this particular strain can efficiently lose the weight. They have measured the weight. This is the control using a different time period study. The time dependent, they have also grown this particular Klebsiella strain. So with respect to the time, the approximate 25% loss in the PVC was observed at around 90 days of incubation of these Klebsiella strains. They have also measured the degradation of PVC in presence of the Klebsiella EMBL strain, and they did the GCMS. So they found that the polyvinyl chloride gets degraded into the small molecules, small carbon molecules such as this. One is this nanodol adipic acid. This is 2 is the another adipic acid, methylosteresters, and this octyl ministate is this is the compound, and so other compound was also reported. As a result, this long chain PVC can convert it into the simpler 6 to 8 carbon long compounds by the action of Klebsiella EMBL01. So this is the overall how Klebsiella act on the PVC. So this is the PVC that can oxidation. First the dechlorination, the chlorine group is removed, and then the depolymerization, then deoxygenases, and ultimately is formed the simple molecules that can enter. Then also different type of leakage, monooxygenases, esterases active. Ultimately this PVC is converted into the acetic acid, and some other molecule. The acetate can be utilized by the bacteria for their metabolic activity. And another way, the monooxygenase, dioxygenases also form the fumarate and some other molecule that can enter into the PCA cycle. So the PVC molecules can be degraded into the small molecules like fumarate or acetate that can be utilized by the bacteria for their metabolic and the growth purpose. Next, last one is the degradation of polystyrene. So the polystyrene mechanism can be the polystyrene is the repeated chain of the styrene. So this can be cut during the degradation mechanism. The polystyrene has to convert into the styrene monomer. So this is the styrene monomer, and different type of enzymes can be used to cleave this polystyrene chain to this form this styrene aromatic group in the styrene, and then ultimately the styrene converts into the acetyl coenzyme or acetyl A, which can enter into the cell for the metabolism, and acetyl CoA participate into the TCA cycle. So this is the degradation mechanism of polystyrene, in which the multiple enzymes are involved like lipase, esterase, peroxidase, P450 oxygenases that can hydroxylate the polystyrene chain for the degradation. And this is the one of the research is Pseudomonas aeruginosa DSM-15071. This particular strain they have isolated from insect larva, and they grown this strain in the film of the polystyrene. This is the control film. This is the microorganism grown on the polystyrene film, and this white is the growth of the Pseudomonas species. It means this Pseudomonas is happily growing in the presence of the polystyrene in the minimal media. They have further observed that this is the SEM diagram, this is the control, and this is the Pseudomonas species. So, with respect to the time, this as bacteria grown onto the polystyrene surface tends to change their surface, make the surface more rough. I mean, the polystyrene gets utilized by this. Also, the serine hydrolysis, they find out how the polystyrene gets degraded. So, they have measured the different gene expression of different genes such as alpha beta hydrolases, esterases, hydrolases, SCT, and also measure the SH serine hydrolases. So, they find out that as compared to the other tested genes, the gene expression level in presence of the Pseudomonas treated cells having the higher serine hydrolases that helps to catalyze the polystyrene by Pseudomonas aeruginosa. And to validate this result, they have used this SH inhibitor, serine inhibitor, with using two different media, nutrient media and this polystyrene containing media. So, this is the control in which there is no effect of the inhibitor. As this concentration of inhibitor increases, the growth is also increasing in the nutrient medium. Once the cells were grown into the minimal medium having the polystyrene, in presence of the inhibitor, as the inhibitor concentration is increases, the growth of the media is also decreases. It means that SH group is involved in the polystyrene utilization by this strain, Pseudomonas aeruginosa. And this is the different research which can tell you that proteinase K induces the biofilm formation of polystyrene. So, in this particular case, there is a bacteria Rhodococcus ruber 208. They have grown onto the polystyrene surface, and then the bacteria biofilm is treated with different type of proteinases, like they have treated with proteinase K and also treated with trypsin. So, as compared to the trypsin, when this bacteria biofilm is treated with the trypsin, the biofilm gets disturbed, while in case of this proteinase K, treated this biofilm of the bacteria with the proteinase K, the biofilm gets more stable, multi-layered. So, this is because of the proteinase K degrade the detachment signaling and leading to form the multi-layered non-dispersed biofilm as compared to this. And ultimately, they have also concluded there are certain kind of enzymes that can be inhibited by the trypsin. As a result, the detachment is occurs. And in this case, it induces some kind of signals, can forms the biofilm related, can reduce the quorum can induce the quorum sensing. As a result, the more amount of biofilm, but the nature of biofilm gets changed. In this case, the nature is multi-layer biofilm is formed by the activation of the proteinase treatment of Rhodococcus rubers. So, these are the different enzymes involved in the polystyrene synthesized in the different organisms like lipase, esterase, hydrogenase, peroxidase that can be reported in different organisms. And the last one is, so far we have discussed the single particular organism, natural condition, degrading the different type of plastic. So, they have modulated the strain to increase the plastic degradation efficiently. For that, they have generated some strain. So, they have used the recombinant DNA technology approach to clone the more than one plastic degrading gene into the vector, and then such vector transform into the E. coli BL21D3, and that recombinant host can be used for the measurement of the plastic degradation. So, this is the one example they have measured to plastic degradation. In that case, this is the empty vector, which is empty vehicle, and in one case they have clone this DGC genes. This DGC is the diagonalate cyclase, and this particular gene increase the cyclic di-GMP, which increase the biofilm formation. So in this one case they have make such kind of clone. Another case they have also develop a clone clone this WSPR genes, and third case they have also make a single gene clone into this factor DH3. So DH3 is the PAT degrading enzyme, polyethylene terephthalate enzymes, and also TF-cut2 they have also cloned this TF-cut2 gene into the vector. Single as well as the double cloning they have made, and also check the polychlorolactone label in this all kind of recombinant strains. So as compared to this DH3 single clone gene, this double gene having the PH3 and DGC gene having the more efficiently degrade this PCL, and if it is WSPR gene is cloned along with the DHR one, has the lower degradation capacity as compared to this. While this TF-cut2 and WS are the highest one, and this CUF2 and DHC has these two has the higher capacity to degrade the polyactic PCL more efficiently up to 80%. So rather than using a single or natural approach, the recombinant approach is the more efficiently degrade the plastic up to 70 to 80% if we clone multiple genes, and such recombinant host can be efficiently degrade the plastic. So rather we can use the natural as well as can also develop a genetic engineering into the strain for efficient degradation. So if we conclude the overall study, we can say that there are different type of plastics are available in the ecosystem, and the different microorganism attack onto the plastic for their degradation. In the plastic degradation process, microorganism adhere to the surface, tends to decrease the hydrophobicity of the plastics, converts into by changing some functional group hydroxylate, and also further increase the hydrophilicity. Such kind of modification occurs in the different type of plastic, makes vulnerable for the degradation by the action of different hydrolytic enzymes secreted or synthesized by the microorganism biofilm or extracellular release from the microorganism. These are the conclusion of oval slides. These are the bacteria which acts in the different type of plastic, produce the different type of hydrolysis enzyme, oxygenases enzyme, proteases that ultimately convert the polymers of the plastic into the monomers, and these monomers can be utilized by the different type of microorganisms for their growth and metabolism. So, thank you.
Good morning, sir. Will you please differentiate between the thermosetting plastic and thermoplastic one more time?
Bolo.
Will you please differentiate between the thermosetting plastic and thermoplastic one more time?
Thermosetting plastic are those which we can heat one time and it gets set, cannot be remolded. It is hard substance.
Okay.
While thermoplastic we can heat it and then again we can heat it, then again it can be molded.
Okay, sir. Thank you.
This is the difference.
Good morning, sir. So my question is, let's suppose there is a persistent plastic biofilm. So sir, can it evolve to a synthetic biome?
Synthetic?
Biome.
Yeah, we can do this one. Even we can also develop different type of nick we can also generate. Consortium we can also generate for efficient degradation. That we can do.
And sir, is there any method of plastic degradation by this biofilm being sustainable?
So far, if we look at this last slide I have given, if we use this multiple organism or multiple clone that can efficiently degrade the plastic, that can be a sustainable approach. But we need to look at whether that approach, how long it will work in the natural environment, whether the plastic, how efficiently it degrades. Because sometimes if we grow the microorganism, in the natural condition, the growth of the microorganism can also be suppressed. So depending upon the environmental condition and the plastic nature, the growth may hamper. So for the sustainable degradation of plastic, we can use in the control condition rather than to the natural condition. Or maybe that not today, but later on the microorganism may also develop such kind of nature, such kind of alteration that may efficiently degrade the plastic. If you look at the history of back, like 20 years back or 25 years back, there is such no microorganism present in the ecosystem because the plastic level was less. But as that ecosystem has the more plastic, so microorganism tends to develop their own adaptation against the plastic, can efficiently degrade. It may be in future the organism can be efficiently emerge depending upon the situation of the plastic.
And sir, one more question is: Is there any such species by removing which it can hamper the degradation process?
Removing the degradation process?
No, sir. Is there any keystone species in this method?
Keystone species?
Yeah. After removal of which this process will get hampered.
So after certain, it normally in this case, some other case as well, if it is the higher concentration of simple or end product will be higher, then the overall growth of the microorganism can also be suppressed. So there are two factors. One is the degrading mechanism. Another one is the how much amount the last or end product is synthesized, whether end product is suppressing the growth or not, that we need to take care about the degradation process.
Thank you, sir.